My name is Soni. I'm a postdoc in the food assurance team, working with Massey University on microwave sterilization project, which is part of a bigger project known as Food Industry Enabling Technologies. Bear with me while I share my screen with you. Yes, so my work focuses on developing methods to validate coaxially induced microwave assisted pasteurization and sterilization. Going back to the history of microwave, Percy Spencer, who was a recipient of several patents, discovered that food can be cooked by microwaving somewhere in 1940s, and with a lot of research somewhere around 1954 Raytheon Corporation had developed, assembled and sold the first commercial microwave. Since then, microwave has had immense amount of applications, and this includes drying, extractions, and cooking of food. It gives quicker results as compared to the conventional heating and also is easy to operate and requires less maintenance. However, there are a little bit of issues associated with the dielectric properties which regulate the heating process in microwave, and it has been known to have non-uniformity and variation in heating. For example, in the image on the right, you can see a thermal camera image of a tray processed through household microwave. So the center portion has received less heat as compared to the rest of the region in the tray. This sort of problem needs to be covered. To know more, we have a manuscript accepted in Trends in Food Science and Technologies, which would elaborate details on this. Now, the technology itself combines long wavelength microwave energy where food in polymeric packages are heated at more than 120 degrees Celsius in pressurized cavities using hot water along with microwave. The graph here shows a time temperature exposure graph uh, in CIMPAS or microwave sterilization unit, and that in a retort. So as you can see, the whole time at 121 degrees Celsius is comparatively much lesser in CIMPAS or microwave sterilization unit. Because of this, though it is a potential sterilization method to eliminate pathogens, it would also have relatively less loss of heat sensitive nutrients as compared to that of retorting. It's important to note that this is a thermal technology from the regulatory point of view as approved by Food and Drug Administration. Now, how CIMPAS works. So this is a schematic overview of the parts of CIMPAS, where you can see there's electrical cabinet here for the control of waveguides, microwave power, and all that. This is the control cabinet from where we can regulate each and every part separately and together. This is the processing vessel for the foods to be pushed in and out. And this is the hot water tank, which operates at about 121 degrees Celsius. And this one operates somewhere at 30 or 65 depending on the regime. This is our warm water tank. How CIMPAS works continued. So the carrier tray with product is inserted into the vessel. From there, the vessel is flooded with warm water, which is at 65 for the preheating regime. That goes on for 20 to 30 minutes. After that, the hot water under pressure is flushed in, which is set up at 121 degrees Celsius approximately. And later on, the microwave power is switched on. Now, under this setup, the carrier tray with food trays is passed through the microwave antenna for a set period of time. Later on, the hot water is flushed back and the warm water, which is below 30 degrees Celsius, is flushed into the vessel to cool down the product. This is how the broccoli puree before and after treatment looks like. You can clearly see that there are these white packaging setup which seals it firm onto the processing tray so that it can undergo the whole process without any rupture. Now, the project itself looks at different methods to validate CIMPAS. For this, the primary method is temperature distribution studies because it is a thermal processing regime and would be dominated by the results produced by the time temperature data. However, the regulatory authorities like to validate a methodology through multiple regimes, and for this we are using chemical markers and microbial inactivation to support the time temperature data. So the time temperature work is being done by the Massey University Engineering Group, and that is through TMI probes and fiber optic probes later on, not yet started. Uh, but the chemical markers being in use here are Browning or Maillard reaction and inactivation of sterilization indicator spores would support whatever data we have. With this, we know all the three methods, or at least two methods, need to be applied for a wide range of food products, ranging in moisture, salt content, and dielectric properties, for us to ensure that if there is a variation in heating, is it significant enough to cause microbial inactivation or not? I would be using this term quite often, which is cold spots. It is less thermally treated areas on the processing trays which receive lower time temperature exposure as compared to the other regions. So this is just an example shown here. So image A is the time temperature loggers or the TMI probes that we are using currently. This is the processing tray of, the food tray of broccoli. And if we place one probe each on two different regions as shown here, and then after CIMPAS processing, if we extract the data out, if the graphs look like this spot has received much lesser heat as compared to this spot. Hence, this is a cold spot. Cold spots can lead to concerns around shelf life and food poisoning. The temperature cannot access more than two regions with this sort of probes that we are currently using. Hence, we have some space limitations there. Also, the cold spots in each food product could be different, which is again dependent on the dielectric properties. Hence, the validation has to be repeated over a wide range. For the chemical indicator, as I mentioned previously, we are using Maillard Browning, where the chemical marker M2, which is a resultant of the precursors D-ribose and amine, which forms amidori compound and then forms the M2. This is our target to understand variation in heating. M2 itself can be monitored by quantification using high pressure chromatography. However, we can also indirectly measure M2 using colorimetry. Before we could apply the measurement of M2 as a chemical indicator to understand variation in heating onto CIMPAS treated trays, we wanted to make sure this works in laboratory conditions. So we used an oil bath setup at 121 degrees Celsius. And this is a capsule where we filled the mashed potato model food spiked with ribose and lysine, and then heat treated it for regular interval of times. This is how it looks like. That's the temperature, that's the sample point 0, and that's the sample point 10. So there's definitely a gradient in browning. It was also confirmed using the average L values, which was significantly different for the last time point, and also the change in total color, which was also significantly different for the last 2 points as compared to the others. So once we knew this method is working at such high temperature, we applied it onto CIMPAS. And before anything, we quantified the M2 as well. For this, we scooped out 1 gram of mashed potato after CIMPAS processing from the hot and the cold spot and quantified the amount of M2 obtained there. So this is a graph where you can see the concentration of M2 at hot gray bars and cold black bars, and the L values on the hot, which is the gray again, and black cold spots. So there is a pattern, there is a correlation, because the cold spot always had higher L values and lesser M2 concentration as expected, and the other way around for the hot spots. So hence the method was validated once more here. Once it was confirmed that this method works to understand non-uniformity in heating, we applied this on CIMPAS process trays in the middle layer, and we mapped 9 spots on each tray to understand non-uniform heating per tray. And after statistical analysis, we found 3 spots for inoculation. One was the cold spot as shown as a blue, and then hot spots are orange squares, and the gray ones are the medium heated spots. we use Clostridium sporogenes spores for the challenge studies. So Clostridium sporogenes is a surrogate for group 1 proteolytic Clostridium botulinum strain, and it is spore-forming, non-pathogenic, and anaerobic microorganism. The D values, which is the time required to reduce 90% of the microbial population at a given condition, was estimated specifically for this strain. And we found the D values to be very different in milky water and that in mashed potato. The difference was about 2.5 minutes, which is significant in terms of time temperature data correlation. Hence, we knew that for every validation method, we need to inoculate these spores in the food and not in any model buffer system. So to get an understanding of the localized heating and inactivation potential, we developed spore pouches. Spore pouches are these barrier bag pouches which are microwave friendly in nature, and they are inoculated with 1 gram of mashed potato model food, inoculated with 10 to 7 Clostridium sporogenes spores, processed through CIMPAS, and then the recovery was done in anaerobic conditions in sheep blood agar plates. These are as results. So the control tray, which was undergoing similar conditions of storage without CIMPAS processing, we could recover the spores on all the 3 spots. But for all the other 12 CIMPAS treated trays, we found more than 7 log inactivation on each spot, may it be hot, cold, or medium treated, which indicated that CIMPAS is able to provide more than 7 log reduction of Clostridium sporogenes. However, we also had to validate if our spore pouches are working or not. So for this purpose, we reduced the hot water temperature to 65 degrees Celsius and processed the trays inoculated with spore pouches in the same way. And the data obtained from the cold spots show that tray 9 obtained better inactivation than all the other trays. And at the same time, we knew that the spore pouches are working quite fine in terms of showing us difference in inactivation. So the summary so far is that we have developed a chemical indicator for identification of non-uniform browning and development of spore pouches to obtain localized inoculation and recovery from these cold spots. And though the cold spots are detected, most of the trays showed all the trays showed more than 7 log reduction of Clostridium sporogenes. And CIMPAS might generate slight difference in heating. However, this is not different enough for lethality achieved at these regions. We have 2 publications so far from this project, and the future work would look at the inactivation potential with lower temperature regimes and high microwave power. It would also cover the effect of moisture content and dielectric properties, and would like to look at the effect of masking of, let's say, fat content in food on this sort of inactivation. Thank you very much for listening.